I'll have two quick questions. The first one is from your first part of the talk. The ERR alpha and gamma seems to be more ubiquitously expressed and not just in the heart. But what I was more interested in is that do you think they act as a repressor of uh, atrial gene in ventricle and vice versa, rather than an activator alone of ventricular gene?
We have done in our experiments where we like mutate enhancers. So we've done experiments. We take the enhancer and we mutate the binding sites, and we've done that for hundreds of enhancers, and almost always the nuclear receptors are activating. So if we ablate the binding site, the activity of the enhancer will go down.
I see. Uh, and the second question about your antisense oligotherapies, I mean, how stable are they once the treatment occurs as compared to some of the conventional siRNAs or other approaches that are present?
Yeah, all of these engineered oligos are amazingly stable. So like we actually in our model, we showed that the mice do not get VT again for over 5 weeks if you stop treatment. Some of these clinical oligos for the liver last for months.
Yeah. Wow, wow. Thank you.
Yeah.
Bill,
Yep.
one question for you is in your comparing atrial and ventricular myocyte, you started at P0 and analyzed at P7 when cells are still undergoing development. The question arises how stable the changes are, or is this snapshot, or if you repeat this, let's say in P30, would you see the same enhancers in transcriptional occupancy or differences in transcriptional activity in atrial and ventricular myocyte, or just during developmental stage you see that?
Yeah, that's a great question. Ideally, we would have done an earlier, later developmental point. We picked those time points for technical reasons, that the data quality is much higher at that time point. But in terms of specific enhancers, we did test several of those enhancers just as individual enhancers. And they do retain their chamber selective activity, into adulthood. But as to, whether all of them do, I can't say that.
So while there is a question from audience, but I can't quite get what it means. Meanwhile, while I can trying to get my neurons to digest that question, do you think that the spatial distribution of calmodulins, I was reading somewhere that 1 and 2 and 3 might not have the same intercellular distributions, and therefore it's not necessarily the total level that may not change, but the spatial distributions are distinct. And do you think there's functional significance to those, the spatial, one being in different parts of the myocytes and so forth, being functionally important?
I guess it's always possible. I think that would be the Achilles heel of the strategy, is that there's not complete redundancy of these isoforms. All we could say is that so far in the mice that have over a 90% reduction of CALM1 in the muscles that we looked, in the tissues we looked at, we didn't see anything bad happening to them.
Yeah.
But there's also the theoretical possibility that.
Fantastic. Bill, now I can read the questions from audience. First of all, let me acknowledge Yibin Wang, who has been a wonderful colleague and an active member of this webinar series. If his microphone could be activated, he could read his question. Otherwise, while his microphone is being activated, Bill Yibin says, "Dear Bill, beautiful lecture and thank you," in capital. My question is how CMYA5, this is cardiomyopathy A5, isn't it? That's what it's.
Yes, that's it.
Yeah. I believe. Interaction with ranitidine receptor 2 is impaired in heart failure and why the expression is not affected, but overexpression of the mini form can rescue the disease?
So the RNA level of that protein is not changed. The protein's a little more complicated because it has many bands on the Western, which we think are actually real because when we do the Western with the knockout, you don't see those bands. So there's many isoforms, and when we do IP with the ryanine receptor, only one of them IPs with the ryanine receptor. So we think there is some protein level processing, although we're still not sure exactly what it is, and that changes in heart failure.
Very good. And then I have the next question from a former colleague and trainee is Professor Swetny Chen. She has two questions. She is from University of Colorado. How ERR affects on cardiac myocyte properties? I guess you meant to say how ERR affects cardiac myocyte properties. Does it regulate the stoichiometry of the sarcomeric protein or channel proteins? What are the targets of ERR?
So Dan Kelly has done most of the work on ERR, and he's shown that it affects both expression of mitochondrial biogenesis genes. That's something it seems to do in many different tissues, not just the heart. And it also regulates cardiac maturation. So it affects, many of the genes that we normally associate with a maturing myocyte, as well as the ultrastructure of the myocyte.
Before we move on to Dr. Dangari, Sydney's second question is, does CMYA5 also regulate the assembly of desmosomes in PS cardiac myocyte, particularly the expression of DSP?
It's interesting. We also study DSP, rhythmogenic cardiomyopathy, but we haven't intersected the two projects to see how one affects the other. I don't know off the top of my head. I wouldn't know like why CMYA5 would affect the desmosome proteins, but so we haven't made that measurement, but it may.
Dan, please.
Yeah, thank you very much, Bill. That was beautiful work, and congratulations. Regarding I just have a couple questions. One regarding your mini CMY 5A rescue. If you continue to look beyond the 28 days, do you see continued improvement of cardiac function, or does it reach a plateau?
So we did this. Our study was 2 months long, and it was somewhat stable between like approximately 4 weeks and 8 weeks. We haven't done a longer term study. We did do a reversal study. So the study I showed you was like prevention, but we have done the next step of adding it after the TAC, and it is effective in that context as well.
And when your studies with regards to the knockout of this gene, you looked at pressure overload. Have you looked at other forms of heart failure?
No, we've only looked at pressure overload. Yeah. So in the knockout itself has only a mild phenotype. Like the fractional shortening goes from our normal, it's 50, it goes to like 40. But in the context of pressure overload, then it drops to like 10. It's a very severe effect of the knockout plus pressure overload.
You've spent a long portion of your career focused on transcription mechanisms that drive transcriptional activation, but kind of following up on the first question, how do you view transcriptional repression in the heart and regulation of GRNs from that standpoint?
I think it's a huge unexplored area. I mean, and I think a lot of that is technical. Like our assays are designed to look at activation, and it's much harder to measure repression. But like we did quite a bit of work on EZH2 and how that is an epigenetic repressor. I think there are many repressors and probably transcription factor mediated repression that is not yet very much studied, and that's in part due to the lack of good assays to study it.
Thanks.